Hi, everyone. I hope that you're all staying healthy and safe at home, and thank you for the introduction, Koei. I also want to say thank you to the dkNET organizers for this opportunity to talk about what we do at Addgene and how we support open and reproducible science. I'll also talk about what materials and resources we offer that could be helpful for the dkNET community and the benefits of depositing as well as how to deposit. So before I dive into what Addgene does as a plasmid repository, I wanted to share a scenario that some of you may have experienced when dealing with acquiring or sharing key research reagents like plasmids. So in this scenario, let's say that we have a researcher who makes a plasmid and publishes it with a statement that the plasmid is available upon request. Then eventually that scientist moves on from the lab and stores that published plasmid somewhere in the freezer. A few weeks or months pass by and other scientists read that paper and request the plasmid. But unfortunately, no one else who is still in the lab remembers where exactly it's stored. Yikes. So this is just one such scenario involving a problem with the traditional way of sharing reagents that can ultimately waste researchers' time, money, and resources. On the receiving end, as a requesting researcher, many of you may have experienced waiting weeks or even months to receive a published plasmid from the corresponding author, or even having to give up and just recreating the materials yourself. For the corresponding author, they have to deal with the burden of keeping track of all of the reagents, authenticating them, storing them, and dealing with the legal distribution as well as the shipping of the reagents. And if these reagents are in high demand, they may not even have the resources to do all these things. So I just want to make a point that centralized reagent repositories are a part of the solution to these problems with sharing reagents. Repositories like Addgene are well equipped to handle the logistics, storage, validation, and distribution of these materials. At Addgene, our mission is to accelerate research and discovery by improving access to useful research materials and information. And we do this by helping scientists easily and quickly share their materials. And so here's how it works. A lab makes a new plasmid, they deposit it for free at Addgene. Addgene performs quality control on the plasmid and makes it available in the repository so that labs from around the world can request and gain access to these plasmid tools. And some of these researchers may make useful modifications to the original plasmids that they received through Addgene, and maybe they'll decide to deposit with Addgene to continue this cycle of sharing plasmids to advance science. So we save scientists time and money by taking care of the logistics of plasmid storage, quality control, and distribution. And for the requesting scientists, we help them quickly find and access the tools they need. In terms of what we offer, we have a large collection of plasmids, including pooled screening libraries and plasmid kits. We also produce ready-to-use viral preps for our most popular AAV and lentivirus plasmids. And we also have a lot of free educational resources online. To give you some stats, Addgene has been around since 2004. And since then, we've shipped over 1 million plasmids to scientists around the world. We currently have more than 86,000 plasmids in our repository from over 4,000 depositing labs, and we've shipped to approximately 100 countries so far. We also have over 500 ready-to-use viral preps available. And before the pandemic, we were shipping around 700 items a day, and that number has reduced due to the academic lab closures in response to this pandemic. But we are still open and shipping, and currently focusing our efforts on helping scientists with the COVID-19 research response, which I'll talk about in a little bit. And so our educational resources, which I mentioned earlier, include our blog, which gets over 120,000 views per month. And many Addgenes contribute to writing these blog posts, but we also frequently have guest blog posts written by scientists in the community. So if you have any interest, please feel free to reach out to us about that. We also have over 30 written protocols and over 10 protocol videos that you can see on our protocol page. And if you are a Protocols.io user, you can also find all of our protocols there. We also have a really great series of free eBooks that cover topics ranging from Plasmids 101, CRISPR, fluorescent proteins, viral vectors, as well as a science careers ebook. And if you're interested, I encourage you to check out these free educational resources we offer. In addition to helping scientists do faster science, Addgene is also committed to supporting reproducible science. And we do this by increasing access, standardizing reagent information, and maintaining quality control of reagents. And here's a really great example of how Addgene has played such a big role in enabling the rapid adoption of a technology like CRISPR-Cas9 by helping scientists quickly distribute and access these plasmids. To date, scientists have deposited over 10,000 CRISPR plasmids from over 900 labs in 29 countries around the world. And we've distributed over 180,000 CRISPR reagents to 87 countries. So this really speaks to how Addgene has enabled the rapid development of this technology by providing access. One other timely example that I want to highlight is how Addgene is supporting the current COVID-19 research response. Addgenes have been working diligently with scientists to acquire and distribute coronavirus-related plasmids for research. We have also created a COVID-19 resource page, which you should feel free to look at. The URL is on the screen. And this is something that this page we've been frequently updating so that scientists studying coronavirus can quickly find the plasmids they need. I also want to mention that some of these plasmids have also been opened up to industry, which means we can make these materials more open and widely used. And another way that we enable reproducible science is by standardizing reagent information. All of our deposited plasmids are assigned an Addgene catalog ID number, as well as an RRID upon availability. And so this makes sure that the researchers can uniquely identify these reagents. All of our plasmid pages are also curated with associated data and information, which I'll get into in a little bit with an example. And each of these plasmid pages has instructions on how to properly cite the plasmid or viral vector so that scientists can give proper credit to scientists can receive the proper credit for the plasmids they made, and so that others who are reading the article can know exactly which reagent was used. Another important factor of reproducible science is proper authentication and quality control of reagents. So at Addgene, for our plasmids, we do full plasmid sequencing for all incoming plasmid deposits. And for the viral vectors that we produce in-house, we perform various QC measures, which may include full sequencing of the viral genomes, endotoxin testing, as well as testing for purity and sterility. So now that I've had the opportunity to talk about what Addgene does, I want to get into some plasmid tools and resources that may be helpful for the dkNET community of scientists. As I mentioned earlier, we have over 85,000 plasmids in our collection, and we have plasmids for all sorts of experiments in many different model organisms. So we have plasmids for studying human genes, for studying mouse, rat, zebrafish, C. elegans, fruit flies. And we also have plasmids that can be used for a wide variety of applications, like CRISPR, chemogenetics, optogenetics, fluorescent proteins, reporters, biosensors, viral plasmids, and more. And you can also find plasmids curated by research fields. And we also work with different research communities who are committed to sharing their plasmids, and we can work with them to create these community collection pages. And I also wanted to highlight the Resolute Plasmid collection for solute carrier proteins. I think that this one could be of specific interest to the dkNET community. And I also want to mention that these plasmids are also available to industry. And we also have a growing collection of plasmids that are, in addition, available to academia, are also available to industry. So if you have interest in browsing these collections, feel free to visit the URL listed at the bottom of this screen. And so we don't yet have a collection page specifically for the dkNET community, but I did put together a table of some popular plasmids, which have been requested either at least 50 to 100 times, that may be relevant to research on diabetes, liver disease, or kidney disease. And another really great way to find plasmids that you're looking for is to simply visit our website on addgene.org. And so now I'm going to give you a brief demo to kind of show you how easy it is to find some of the tools that you might be looking for. And so hopefully you can see my screen here. And so this is our home page. And I just wanted to mention that we recently updated our website and the search function to make it easier for you to find the reagents you're looking for. And I'm going to go through, as I mentioned, a few examples. So let's start off with something simple, like let's say I'm looking for something for a plasmid that's related to insulin. And so I see that there's 170 plasmids that are available related to insulin. And I see that, this plasmid looks interesting. It's pro-insulin nanoluciferase in PLX304. And the purpose is luminescent reporter of insulin secretion. So I can go and click on this. And I wanted to take an opportunity right now to highlight some of the key information that is listed on each of these plasmid pages. So at the very top, you can see the full name of the plasmid, pro-insulin nanoluc PLX304. You can see the purpose, the depositing lab, and if this plasmid was published in an article, you can also click on this link to view the article. And then we also have to the right, you can view the plasmid map. This plasmid map is powered by Snapgene. We have a partnership with them. And you can also retrieve either the image or the full sequence, either in GenBank or Snapgene format. And then as you scroll down, you can see some key information about the plasmid, which includes information about which vector backbone was used, and information about the gene and insert, and if there are any mutations that were made, it's listed here, as well as the entree gene and the aliases, so that it's easier to find these specific plasmids. In some cases, it'll also list the promoter used. And another thing I want to highlight is the article citing this plasmid section. So if you click on this, you can view articles that have cited the use of this plasmid. And so this is, again, not only giving credits to the scientists who made this plasmid, but it also gives researchers who are looking through the information about this plasmid on the Addgene page to find out how this plasmid was used in other studies. And then finally at the bottom, as I mentioned earlier, we have instructions on how to properly cite this plasmid. And so this makes it easy for the researchers to simply copy and paste this phrase into the materials and methods section. And it includes the name of the plasmid, the name of the scientist or the PI, as well as the catalog ID number and persistent identifiers, including the RRID. And so let's move on to another example. Let's say that I'm studying hepatocytes in the liver. And so when you search for any term, not only will you get a list of plasmids, but then it also may find relevant educational resources. So this lists some blog posts that mention hepatocytes. And so this can be another way of finding useful information on the Addgene page related to your research. And so I'm going to click on plasmids. And another way to more quickly find what you're looking for is you can also, there's these filter buttons. And so let's say that I'm studying hepatocytes and I only want to look at mammalian expression, something intended for mammalian expression. And I'm specifically studying the mouse model. And there's other additional filters I can use. Let's say for some weird reason I only have kanamycin available in my lab and at the moment, and so this is what comes up. And I find a fluorescent marker for hepatocytic lineages. And you can also search for even more specific things. Let's say that I'm looking for a specific promoter. You can even type something as specific as, let's say that I'm looking for the plasmid that contains the TBG promoter. And so here we see that there are 8 plasmids that match this term, but then there's also viral preps. And so I'm going to click on this. And we see that there is this AAV8 viral prep that's available. And so when I click on this link, it'll show the plasmid that the viral prep was created from. And so we see that this is an empty AAV vector with the TBG promoter, which is what I was looking for. And then when you scroll down, you can see the information about the plasmid. But then when you scroll down further, you can see specific information about the specific viral prep that you may be interested in learning more about. And so here it'll provide some information about the purpose of this viral prep. It'll also have some information about the titer and volume that you'll receive, and other information here. And again, it'll also have the information at the bottom of how to cite this plasmid or how to cite the specific viral prep. So I wanted to also highlight, I mentioned earlier that we have a lot of different collections. And so I just wanted to go through one case where, let's say that I'm a zebrafish researcher. And so, as I mentioned, we have a lot of plasmid collections and science guides that are really helpful in guiding you through, what experiments you might want to run. And so let's say that I'm a zebrafish researcher, and this page is nicely organized, and it lists a lot of different specific tools that I can use in zebrafish. And it also has these tables, and this table lists has 189 entries that are related to zebrafish related plasmids. And I can look at the purpose of these plasmids here, and look at what is the gene insert in this table. But then you can also search the table, and let's say that I want to look for insulin, something related to insulin. And so here there's a plasmid that allows specific and conditional cell ablation of beta cells in the zebrafish pancreas. And the plasmid includes an insulin promoter driving CFP nitroreductase to ablate only those targeted cells. And so hopefully this demo has given you an overview of how to navigate the website, and how some of the tools you can use to find the plasmids that you are looking for. And so now I'm going to switch back to the presentation. Let's see. Okay, so hopefully you can see my screen here again. And so I just wanted to mention that if for some reason you need help finding something in particular, or you have any technical questions, we also have a really dedicated team of Addgenes who do customer support and scientific support to help you answer any questions that you have, or to help you find what you need. Additionally, if you read a paper with a really useful plasmid tool that does not exist in Addgene's repository, you can also fill out our Suggest a Plasmid form, and we can contact the corresponding author about getting those plasmids deposited. So now I'm going to talk a little bit about the benefits of depositing, and why and how depositing works. So I hope that you will consider depositing, and one reason is that maybe the image to the left portrays what your freezer looks like, and the photo on the right is what our freezers look like. And there are many benefits to depositing your plasmids with Addgene. And so, as I mentioned before, it can help you save time and money, so that instead of handling plasmid requests, you can focus on what's important, which is your research. We can help you stay organized, so that you don't lose your plasmids or cloning information due to lab turnover, or if there was an unfortunate freezer meltdown. We also help to increase citations so that you get credit for your work. And of course, you're sharing with the research community. And we also have a rewards points program. And the best part of it all is that it's easy and free to do so. And so the Plasmid Deposit Process involves a few key steps. The first one is to get us the plasmid information. And so you can start the Plasmid Deposit Process by either going online, or you can email deposit@addgene.org. And then the next step is that Addgene takes care of handling the deposit agreement and MTA with your organization's tech transfer office. Then once that has been approved, then we'll ship you a prepaid deposit kit so that you can ship the plasmids to Addgene. And then we perform quality control, and once everything checks out, your deposit is complete and your plasmids are made available at Addgene. So in terms of what sort of deposits we accept published plasmids, pre-publication plasmids, and useful unpublished plasmids. So when I mention pre-publication plasmids, What I mean are plasmids in which you've described, and perhaps you're writing a manuscript that's not published yet. And one of the benefits of depositing pre-publication is that you can start the deposit process and get all of your information into Addgene's database, and also get the quality control done ahead of time. And then you have the option of putting it on hold, so that as soon as your paper comes out, these plasmids are immediately available to researchers through Addgene. And then, yeah, if you're really into the spirit of open and rapid sharing materials, you are welcome to deposit useful unpublished plasmids. And again, like some of the information that we need to get this into the database is, the plasmid name, the backbone, insert, antibiotic resistance, some basic information about the plasmids. The information that you see on those plasmid pages, basically. The publication number, so if it's published, you can provide the PubMed ID or the bioRxiv DOI link. Any sequence data or plasmid maps are always helpful. Any format will do. You can even send us a basic FASTA format file. And of course, if you have any protocols or other useful documents that we can add to those plasmid pages, that's always helpful. And so once we get that information into our database, Addgene works with the tech transfer office at your institution to ensure that the proper material transfer agreements are in place. And so basically, Addgene does most of the work here for you by directly communicating with your TTO office. The only thing you'll usually have to do is, if you're the PI, you'll need to sign a one-time deposit agreement, which gives Addgene permission to distribute your plasmids on behalf of your lab and your university. But then after that, any time that a researcher requests and receives your plasmid, Addgene takes care of the MTA for you. And once we've received the data, Addgene will send you a deposit kit that includes barcoded stickers for labeling your samples, as well as a prepaid shipping box to ship it back to us. We prefer that you send your samples in a DNA format, if possible, and we'll take, all we need is 10 microliters of mini prep-quality DNA in a tube. And we also do take bacterial streaks, but if you're planning on doing it that way, just let us know ahead of time. And once the deposit kit has arrived back at headquarters in Watertown, we will proceed with sample storage, which includes bacterial transformations, and then storing the plasmids as bacterial glycerol stocks in triplicate. And then we also perform quality control, which involves performing full plasmid sequencing to verify these plasmids. And if there is a case where there's any discrepancies in the sequence, or we have any questions, we'll proceed with you, or we'll check with you, and before we proceed with depositing or making the deposit process complete. And then if everything checks out, then the deposit is complete and the plasmids can be available on Addgene. And so once the plasmids are available, if you're a new PI, you will get your very own Addgene PI page, and it'll have the name of the PI on top, and then also list all of the plasmids that have been deposited. Another benefit of having your plasmids in Addgene is that you can also view requests for your plasmids by logging into your Addgene PI account. And so this is a really great way to find potential collaborators, or just to know how many times your plasmids are being used and who's using them. And as a small thank you, we also have a Addgene rewards points program. So every time you get 15 requests, you get one free plasmid. And I also wanted to mention that in addition to PI pages, we also have organizational pages at Addgene. And so here's an example of what the NIDDK page looks like, and it'll list, by default, it'll list all of the PIs that are located or associated with the specific organization. But I also wanted to mention that we do have customized organizational pages at Addgene. And so in this image, you can see that that this page is like more customized. So it has some, a paragraph, an introductory paragraph in the beginning, and also has links to relevant pages. And then it also has stats, customized stats about the organization. And this is really for, groups or organizations that are very committed to encouraging or requiring their scientists to deposit. And I think that, this encouraging of depositing within your group is a really great way to advance science within your community or field of research, because then all those research reagents are much easier to find in a centralized location and then accessible by your colleagues. So I hope that after this presentation, you've gained a better understanding of the importance of reagent sharing through repositories, and that you've learned about some different plasmids and resources available through our repository that might be helpful for your research. Thank you very much for listening.